Hello, welcome to Biology for You. In this video, we are going to describe chord curves. In the series of videos, we have been discussing different concepts in molecular biology. In this video, we are going to discuss what exactly are the chord curves and why, what is the different terms like genomic complexity, renaturation, kinetics, and like that. And here, coming back to the first concept, what are chord curves? Basically, these chord curves are the graphical representation of the renaturation kinetics of an organism. And basically, these renaturation kinetics are the expression that is used to understand the genomic complexity of an organism. Basically, what is genomic complexity and renaturation kinetics? So first we need to understand these terms. So what is genomic complexity? Genomic complexity is nothing but the total length of different sequences of the DNA of an organism. So this is expressed, this genomic complexity, that means the total length of different kinds of sequences present in the DNA, will be expressed in the terms of renaturation kinetics, which again will be graphically represented in the form of chord curves. So here, basically, what are renaturation kinetics? Renaturation kinetics are the measure of genomic complexity, as we already discussed. And basically, if the genomic DNA will be subjected to denaturation, so if the temperatures are higher than the melting temperature, when the DNA is subjected to higher temperatures than its melting temperature, the DNA uh, will be denatured. That means the two strands of the DNA will be separated by breaking the hydrogen bonds between them. So after denaturation of the DNA of an organism, again the renaturation conditions will be brought back. That means the temperatures will set low and renaturation will happen. That means again the hydrogen bonds will be formed between two strands of the DNA. But here it takes some time for renaturation of all its DNA present in the sample. It takes some time. So here, in the intervals, we are going to take the calculations. The quantity of the DNA that present in the that left in the single stranded form. So we'll take these calculations at every interval during its renaturation. So here this will be expressed in the equation like dc by dt is equal to kc square. So here C indicates the content of the DNA or the single stranded DNA present in the sample at the time t. So whenever you take an observation, the time will be t, and the content of DNA that left single stranded will be C, and here K indicates the second order reaction constant. As renaturation kinetics follow the second order reaction, the K is a constant for that. So if you generally, this renaturation kinetics will be expressed in the form of chord curves, as we already discussed. And here the time half, that means the half time for the renaturation, or the time taken for half of the DNA to renature, will be considered as t half. So this is mostly calculated to understand the renaturation kinetics of the DNA. So let us rewrite the reaction equation like this: C by C naught is equal to 1 by 1 plus k cot. So here C0 indicates the content of DNA at initial time. That means the total amount of single stranded DNA will be expressed in the form of C0. And C is the, as already discussed, content of DNA that left denatured during the time T. So here C by C0 express the proportion of the DNA that left single stranded to the proportion to the DNA at the initial stage of the experiment. So before its denaturation, the total DNA versus the content of DNA that is still left denatured during the time T. So this expression will describe this. So here at the time T half, that means the time at which half of the DNA is renatured. So at the time of T half, this expression will be half of, that means this is 0.5, and the product of C0 into T will be cot half. That means T will be at T half, expressed as T half. So here we can also write the reaction simply. So here cot half will follow this. That means the time taken for half of its renaturation will be inversely proportional to its constant. So this is the expression mostly used in the in plotting cot curves. So here cot curves will be more or similar to like this. So if you consider different organisms, let us consider simple organisms first. So the cot curves will be like this. These are simple sigmoid curves, and here on x-axis we can see the cot values, and on y-axis we can see the single-stranded DNA that left denatured. That means the single-stranded DNA at time t. So here these type of sigmoid curves will be indicated by the simple organisms. So let us consider a synthetic DNA polymer, that is poly-U, that will be very much simple. So the cot values on x-axis showing very much low, and here this may be T4 bacteriophage, and this will be E. coli. If you consider these three examples, the cot curves will be more or less similar to this. The shape of the curve is sigmoid, and they follow the order. So from simple to much complex than the simple. So if you consider a higher eukaryote like human being, the cot curve is not a simple sigmoid curve. Instead, it follows. So if you take cot curves of a complex organism like human being, so the cot values you take on the x-axis, and on the y-axis the single-stranded DNA content will be taken on y-axis. And if you plot a cot curve, these kind of regions will appear. So it is no more a simple sigmoid curve like simple organisms. And here the time taken for this peak here indicates the highly repeating sequences. And this peak indicates the moderately, and this indicates the unique sequences. As we already discussed in the previous So the chord curves are dependent on its complexity of the genome of an organism. So here these kind of chord curves will observe due to the complexity of the genome of a human being, because due to the presence of different lengths of the different sequences. That means uh, some of the sequences in the human genome are highly repetitive, so they highly repetitive sequences will take lesser time to renature. So the probability of colliding of two separated strands for a repetitive sequences will be lower than the time taken for colliding. Probability of colliding of two strands of the having which are having unique sequences in them. So due to the presence of repetitive sequences, the renaturation will be high. They have high complementarity between two strands and they take less time to renature. So this these highly repetitive DNA will renature first, and then the moderately repeating sequences, then the unique sequences, or the non-repeating sequences, or the coding portion of the genes. So here basically these highly repeating sequences, for example these are the satellite DNA, uh, and also mini satellite, microsatellite will also come under this, and these sequences will repeat um, much higher time than the other sequences. They may repeat from 50,000 to 1 lakh times, or maybe higher than that. So this moderately repetitive DNA, the popular example for this is transposons. So this moderately repeating sequence DNA will be repeated as many times as 5,000 to 10,000 times, more or less similar to that. And these unique sequences that are coding portion, mostly the genes, will be expressed uh, with minimal number. That means that may be 1 to 5 sometimes. So these kind of variation will be observed in the case of chord curves. So here the high repeatedness will decrease the time taken for the renaturation. So that is what the chord curves are. So when you take in the previous example, if you consider that polymer of synthetic polymer we have taken, that is having high complementarity, due to which it renatured first in the previous example of this sigmoid chord curves. So likewise, even in the human sequences, highly repetitive sequence uh, will renature first. This is all about the chord curves, and hope you understand. And if you really like the video, please like, share and subscribe to my channel.